Good morning. I am Shivaram Krishna, lecturer in botany, Government Degree College, Puttur. Today I am going to present a topic on physiology of flowering and photoperiodism. It is a it is the topic of fourth semester students for the fifth unit. Plant hormones and the environmental cues. Environmental cues means the signals. These are the key factors which induce the flowering in the plants. Generally, plant hormones we discussed auxins, gibberellins, cytokinins, abscisic acid, and ethylene. From that, auxins, gibberellins, and cytokinins and ethylene are also important for the flowering. And environmental cues, that means low temperature requirement and particular photoperiods. These are the environmental signals which are required for the flowering. Next, environmental cues, that means signals, are the day length, that means photoperiod, and temperature. These are the very important factors which induce the flowering in plants. Generally, the plant switches from vegetative state to reproductive state. That means generally a plant which is growing in the vegetative, it switches its mode to the reproductive phase. That means production of flowers, fruits, and the seeds. Next, generally the switching from the vegetative to reproductive based on the transformation of vegetative shoot apical meristem into a reproductive structure. Generally, shoot apical meristem has to change into a specific reproductive structure. That means the shoot apical meristem always it produces the leaves. But in case of the reproductive structure, that means it have the thalamus, sepals, petals, stamens, and gynoecium. These are the reproductive parts. Next, the transition of vegetative to reproductive phase is marked by the frequency of cell divisions occurred within the central zone of the shoot apical meristem. Generally, when you go through the anatomy of the shoot apical meristem, we can find the central zone. In the central zone, there is a cell divisions occurred. When the plant transits from the vegetative to reproductive phase, generally there is a large amount of cell divisions has occurred in the central zone of the shoot apical meristem. The events which are taking place in the apical meristem which lead to the production of flowers are collectively called as floral evocation. Generally, floral evocation means the apical meristem in which all the changes that leads to the production of flowers, generally they are collectively called as floral evocation. Next, the photoperiodic induction. Initiation of flowering in a plant after the exposure of a light for a required period of time, that is called as the photoperiodic induction. Next, the photoperiodic stimulus is perceived by the leaf. As a result of floral hormone called florism is produced in the leaves, which is translocated to the apical tip, subsequently causing the initiation of floral primordia. That means leaves will generally perceive the photoperiodic stimulus. Generally, it is converted the stimulus into the florism, a hormone called as florism. Then the florism, which is produced in the leaves, that is translocated to the apical tip, and then that leads to the formation of the floral primordia, that means floral meristem. Next, experiments. Generally, in the cocklebur plant, that means Xanthium pennsylvanicum, generally it is a long day plant which proved that the photoperiodic stimulus perceived by the leaf. Generally, leaves are the essential parts which perceives the day length. Through the experiments, it proved that leaves are the parts which they perceive the photoperiodic stimulus. In this experiment, one plant that is defoliated and other plant with having the leaves. Generally, the plant which is defoliated and exposed to short daylight, there is no flowering. And flowering is occurred in the plant which all the leaves are removed except one. That means in the second case, removed all the leaves except the one. But in the first case, there are no leaves, complete defoliation. From this experiment, came to know that leaves are the parts which perceives the photoperiodic stimulus. Next, grafting experiments also supports leaves are the parts which perceive the photoperiod. Cocklebur plant is kept under long day conditions where no flowering occurs. Generally, it is a short day plant, but there is no flowering in long days. A single leaf is exposed to short day condition is grafted to the long day plant. Generally, single leaf is exposed to short day condition, then it is grafted. As a result, flowering is occurred. That means leaves perceives the short day condition. Through the grafting, the stimulus is translocated into the grafted plant. These two experiments proved that leaves perceive the photoperiodic stimulus. Next, there is the first figure is shoot apical meristem. It is the anatomy of the shoot apical meristem. In the vegetative phase, we can see the vegetative phase of the meristem. Just a leaf primordia is observed. In the next figure, we can see the floral meristem. That means vegetative meristem, how it changes into a floral meristem. That means reproductive meristem. We can find that the floral parts in different orals. These are the figures. Next, photoperiodism. What is photoperiodism? Photo means light, periodism means the amount of the period. The plant, in order to flower, require a certain day length. That means the relative length of the day and night, which is called as a photoperiod. Every plant, it requires certain amount of the day length or certain amount of the night period. That it is called as the photoperiod. Next, the flowering response expressed by the plant to the photoperiod is called as photoperiodism. That means when a plant is exposed to a particular photoperiod, that means either short day or long day, it expresses the flowering response. That phenomenon is called as the photoperiodism. That means every plant, after perceiving a certain day length, it shows the response. For example, short day plant, it is when it flowers only during the short days, that means in the winter season. The plants which flowers within the long days, that means in the summer, after it obtains the long photoperiods, it will show the flowering response. Next, discovery of photoperiodism. The concept of photoperiodism was given by the Gardner and Allard in the US Department of Agriculture while studied the flowering in Maryland mammoth, a variety of tobacco plant, in the 1920s. Maryland mammoth, it was a mutant tobacco that did not flower during the summer like wild type. Generally, it does not flower in the summer, just like the wild plants. In the controlled experiments, that means manipulated the amount of the light and dark, they discovered that flowering only occurred if the length was 14 hours or less. That means generally it does not flower in the summer. Due to the mutations, it becomes a short day plant. That means when it perceives the 14 hours or less amount of the time of day period, then it starts the flowering. They call the Maryland mammoth a short day plant because it requires light period shorter than a critical day length to flower. Okay, the Maryland mammoth plant is considered as the short day plant, that is a mutant variety of the tobacco. Next, hence it has been concluded that the relative length of the day is a prime factor in the flowering response. We can see the flowering of Maryland mammoth. Generally, Maryland mammoth is a mutant tobacco of right when compared to the wild tobacco, which is in the left. Generally, with the white we can see the flowering, but in case of the Maryland mammoth, there is no flowering. It grows very large, but there is no flowering during the summer condition. Both plants were grown during summer in the greenhouse. Generally tobacco plant has to flower during the summer, but the Maryland mammoth on the right side does not flower within the summer. Next, Gardner and Allard, the 1920, due to their experiments, they given the concept of the photoperiodism. Generally, after the experiments with the Maryland mammoth, they given the photoperiod as one of the phenomenon which cause the flowering response in the plants. Later, Sumner and Bonner in 1938, they done the experiments and concluded that that dark period is also a determining factor in photoperiodism. Gardner and Allard, they given the day period is the important for the flowering response, but Hammer and Bonner experiments, they concluded that dark period is also an important factor which expresses the flowering response in the plants. Okay. Then the critical day length. Critical day length means the period of the day that is specific in length for any given species that appears to initiate the flowering. That means the period of the daytime which is specific in length, that means a particular hours are required for a particular species to initiate the flowering. Next, depending upon the duration of photoperiod, plants are classified into the three types. One is the long day plants, short day plants, and the day neutral plants. Here, in the first picture, you can see the long day plant. When the day length is below, there is no flowering is found. But in case of the day length is more, flowering response has shown. Next, in second case, in the second figure, you see that in the middle one, the short day plant, when the day length is less, that means night time is more, flowering is occurred, but there is no flowering in case of the above the critical photoperiod. There is no flowering in the short day plants. In day neutral plant, there is no effect of photoperiodism. It flowering always occurs. That's why they are called as the day neutral plants. Next, on short day plants and long day plants flowering, we can see that in the Maryland mammoth tobacco, here within the short days only it blooms when compared to that of the light hours. When the light hours is more than 14 hours, it shows the flowering. In case of the when the light is more, there is no flowering. In case of that, henbane, a long day plants, it requires in case of the light amount of the light availability is more. In the first figure, you see that in henbane, light amount is available more than 14 hours. Flowering response occur. In other cases, there is no where the dark period exceeds more than 14 hours. There is no flowering response, and the henbane plant remains in the vegetative phase. Next, short day plants. Generally, simply it is abbreviated as SDPs. That means short day plants. The plants which require a short daylight period, that means usually 8 to 10 hours, and a continuous dark period of about 14 to 16 hours for flowering. These plants are called short day plants. Next, in short day plants, the dark period is critical and must be continuous. That means the dark period not only lengthy and it should be continuous. If the dark period is interrupted, even with a brief exposure of red light, there is no flowering occurs. That means even though we provide the short day for a short day in the short day plants, the dark period is exceeded more than 14 hours. But 5 to 10 minutes of light interruption is there in the 14 hours of darkness. There is no flowering occurs. That means from this what we can conclude, the length of the night period is very important for the initiation the flowering response in the short day plants. Some are the short day plants, that is Artemisia, that is Ambrosia artemisifolia, Chenopodium album, Chrysanthemum, Cosmos sulphureus, Euphorbia pulcherrima, Glycinmax, and Nicotiana tabacum. These are some short day plants which initiates the flowering only during the short days. Next, we can understand with this figure the short day plants. In the first figure, we can see that critical duration of darkness. That means shaded in the dark, and light is shaded in the yellow color. Light when the light phase is exceeded, the dark phase is less. In case of that first figure, you see the plant, there is no flowering. In the second figure, in the middle one, you see the dark period is more than the critical duration, and the light period is very short. That means short days. Here, the flowering response has been observed in short day plants. In case of the last figure, third one, you see that amount of the night is more than 14 hours, but the dark period is interrupted with a flash of light for a few minutes, but there is no flowering response. From these experiments, we can conclude that the day length, along with the day length, continuous night without any interruption is also an important factor to initiate the flowering response in the plants. Next, long day plants. Long day plants generally short abbreviated as LDPs. The plants which require a longer daylight period, that is usually more than 14 to 16 hours, to show flowering response are called as the long day plants. That means generally these long day plants will flower during the long days availability, particularly the summer period. Next, light period is critical, and interruption of continuous daylight with the dark conditions effectuate the flowering in LDP. But in the light period is critical, and interruption of continuous daylight with the dark conditions effectuate the flowering in LDP. That means there is no problem of providing the darkness within the daylight period. Next, these are the some plants, the Beta vulgaris and Dianthus superbus, Hyosemus niger, Lactuca sativa, Spinacia oleracea, Raphanus sativus, and these are some of the long day plants. Next, this is the long day plants experiment. That means you see the first figure, the daylight is exceeded than the critical daylight. That's why the flowering response has been occurred. Next, in the second figure, the amount of daylight is very short, and the amount of the night time is more. Then there is no flowering response in long day plants. But in the third figure, there is a interruption within the dark phase. But even the night break with a flashlight is given. When the night break with the light is given, it shows the flowering response. Next, the difference between the short day plants and long day plants. Here in short day plants, they need a photo period of less than critical day length to produce the flower. That means they require a photo period less than the critical day length. But in case of the LDPs, plant require a photo period more than the critical day length to produce the flowers. Next, interruption in light period does not inhibit the flowering. But in case of this, interruption in light period inhibits the flowering. Next, long dark period is critical for flowering. In case that long dark period is not critical for flowering, here in case no flowering occurs if the dark period is interrupted midway by a flash of light. Here flowering occurs if the dark period is interrupted with a flash of light. Here no flowering occurs under alternating cycles of short light and long dark periods. Next, here flowering occurs under alternating cycles of short light, still shorter dark periods. This is the comparison of STPs and LDPs. Next, day neutral plants, that means DN is day neutral plants. Plants show flowering response irrespective of their critical day length, and, also called as intermediates or photoneutrals or day neutrals. Then that means the day neutral plants does not depend on critical day length periods. That means always you can see the flowering. For example, tomatoes and cucumbers, these are some of the day neutral plants. Then you can understand the day neutral plants. See the geomis, Gossypium arboreum, Cucumis sativus, Helianthus annuus, Impatiens balsamea, Mirabilis jarapa, and Lycopus canesculentum. These are some of the day neutral plants. That means irrespective of their day length, they show the flowering response always throughout the year. Next, some plants flower after exposure to short days followed by the long days, generally called as short and long day plants. That means flowers require a short days that should be followed by long days. These plants are called short and long day plants. Example is Iberis, Durandi, and Sickle cereal. Next, some plants flower if they have been first exposed to long days followed by the short days. They are called as long and short day plants. That means two periods are required, long days to be followed by short days. That's why long short day plants called as long short day plants. Example, Calonche and Sestrum nocturnum. These are the examples of the short long day plants and the long short day plants. Next, phytochrome. Generally phytochrome means it is one of the pigment, like the photosynthetic pigments. Phytochrome is one of the pigment which initiates the flowering response in the plants by perceiving the critical amount of the day, and that means photoperiod should be perceived by the leaves. Already we know in the leaves there is a special type of pigments exist that are called as the phytochromes. Generally phytochrome is a biological pigment, proteinaceous in nature. Its main thing is to absorb the light. That means particular photoperiod. The amount of the photoperiod is calculated with the help of phytochrome. Generally, it is found in some plant cells that has been proven to control the plant development. It is a non-photosynthetic and a photoreceptor pigment. It is non-photosynthetic. That means it is not useful in photosynthesis, but only it receives the light as a photoreceptor, but it calculates the amount of the photoperiods. Next, here there are the two types of photophytochrome pigments exist. That is, the one is red light and one is fair red light absorbing one. Red light is generally 660 nanometers, generally called as PR, and fair red light. Fair red light is generally 730 nanometers. It absorbs. When PR form of the pigment absorbs red light, it is converted into PFR form. That means there is interconversion is observed within the PR and PFR forms. That means in the light, what happens? When the 660 nanometers red light perceived by the phytochrome, the PR is converted into the PFR form. Similarly, when PFR form of the pigment absorbs the fair red light in 730 nanometers, it converted into the PR form. That means the interconversion is occurs. PR converts into PFR, PFR converts into PR form. The PFR form of the pigment gradually changes into PR form in the dark. Generally, whatever the PFR formed, the pigment accumulated, it slowly converts into PR form during the dark. PFR is the physiological active form of the phytochrome pigment. Next, we can see the interconversion, and the previous day we discussed the same thing, that PR conversion into PFR. Already I told red light, the 660 nanometers, the PR converted into the PFR. In the same way, in fair red light at 730 nanometers, it absorbs the fair red light, then converts it to the PR. That means interconversion is possible. Flowering in SDPs, short day plants. That means the phytochrome is linked to the flowering response in the plants. Short day plants flower when the night period is long. Already we know that short day plants means daylight is very less and night period is more. That's why in the short day plants flowers when the night period is long. In daylight, phytochrome absorbs the red, that means PR form, which immediately converts into the phytochrome, that fair red light PFR. The conversion actually only requires a brief exposure of red light. In the dark, the PFR is slowly converted back to the PR form. That means a long night means that there is a long time for the conversion of PFR to PR. Next, on the short day conditions, that means long nights, at the end of the night period, the concentration of PFR is low. Why? Because the long night, the PFR converts into the PR. That's why the amount of PFR is very low. That low PFR concentration triggers the flowering response in the short day plants. That means the short day plants, they require the low amount of PFR to initiate the flowering. Next, in flowering in long day plants. Long day plants flower when the night period is short. That means in the daylight, the PR is converted to PFR. That means the PR is converted to PFR. That means accumulated large amount of the PFR. But night time is very less. That's why in the night time, the PFR breakdown, that conversion into the PR form. That means here large amount of PFR concentration will be found. High concentrations will be found. Why? Because night time is less. In case of the long day plants, they have the very short nights. That's why in the short night, the PFR concentration will be remains more. Why? Because night time is very short for interconversion. That's why high amount of PFR will be observed. That high PFR concentration is essential for the triggering of flowering response in case of the long day plants. Next summary we can understand with the help of this summary of phytochrome, entire phytochrome. Generally, what happens during the red light, that means during the daytime, there are the in the light spectrum there are the red light and far red light. That means there are many nanometers are there, from the 660 nanometers, 730 nanometers. 630 nanometers, 660 nanometers is the red light, and 730 is the far red light. The red light, that PR builds up, then what happens, it immediately converts into PFR. That As a result, a large amount of the that low amount of the PFR in the night time, it leads to the activation of the florism. The florism leads to the flowering response in short day plants. Likewise, in the long day plants, the high concentration of the PFR is observed after the short nights. Then the high concentration of PFR triggers the florism activation. The florism activation leads to the flowering response. This is the summary of the phytochrome and the flowering response. Okay, thank you. Through this topic, you understand the importance of the day period, that means photo period, as well as the phytochrome importance in the calculation of the amount of the day length and as well as the night time, triggering the flowering response. As a result, with the help of the photoperiodism and the phytochrome pigments, the plant switches from the vegetative to reproductive phase, where the reproduction is the key event in the flowering plants. Okay, you understand in a better way. Thank you.